First, I have to thank the organizer to invite us to give the presentation to introduce you about our vaccine center as well as the adjuvant platform technology we developed. Okay, thank you. So today I'm going to talk about another adjuvant we discovered by just protein. The previous one is the lipoprotein. This one is toxin, the receptor part of one of the toxin causing the Clostridium difficile associated diarrhea, so-called the toxin A. The receptor is very interesting, so I'm going to go through those for you and identify the so-called potential as an adjuvant. Before that, I'm going to talk about the vaccine center. In fact, I was worked with Sanofi before, and I was recruited back during the SARS time. You know, that Taiwan is one of the so-called problem area, and I was there to try to establish the vaccine center for them. And we have a 4 major mission: is trying to establish the facility as well, recruiting people or training people to work on the vaccine development. Second is setting up a cGMP facility so we can produce clinical material to go for clinical trial, as well as a place to manufacture the BCG as well as antivalent. One of the things you may not know, Taiwan have 4 very toxin, poison snake, and they have to have the antivalent against those snake for the farmer. So we have to produce the material for the Taiwan CDC. And then also have a capability to respond to Taiwan government in case they have a request for emergency production. Just like the H1N1 that time, we produced 4 lots, but we didn't have to use because other big pharmaceutical company produced a lot. So Taiwan government just purchased from the pharmaceutical company. And then, of course, the vaccine center, then we train a lot of people. One of the PI is Dr. Ling, show you one of their programs developed. And we fully so-called small biotech. We have so-called cGMP and different platform to produce cGMP grade material. And then we have a different platform technology to develop a vaccine. And so we position ourselves in the middle stream because we can collaborate with other people in upstream to make clinical lot and then go to clinical trial and then prove of the concept in human. And the facility, we have a so-called the cGMP manufacturing, and then we have around 15,000 square foot research area for research and development. And then the cGMP is around here. And here just show you the floor plan so we can produce bacteria vaccine as well as wild vaccine. And then we can do so-called filling and packaging. And we have a cGMP warehouse and animal facility, and then the BCG manufacturing facility. So it's fully capable to handle from research all the way to phase 1 to clinical trial in the vaccine center. Here now I switch gear to talking about the Clostridium difficile associated diarrhea caused by this so-called toxin A. And I'm sure everyone knows if you have been surgery and the doctor will give you some antibiotics, and then this is the problem come when they give you antibiotics, then the Clostridium difficile will become dominant and then will produce the toxin and causing diarrhea, in particular for those elderly as well as the young patient. So CD is gram-positive and produce the toxin A and toxin B. The toxin A is the major toxin causing the diarrhea. And also, the people normally will recover, but some of the people will continue subject for the reinfection. And the major problem for the so-called the CD, this bacteria, is antibiotic resistance increasing. In Taiwan, is in the last few years is almost 40 to 50% increase when the isolate, like clinical isolate, is already multi-drug resistant. And in fact, US CDC recognized this CD of this bacteria becomes the number 1 or number 2 most important bacteria to have the vaccine. Of course, the MRSA is the strep aureus is the number 1. This definitely is the number 2 to have the vaccine or new antibiotics to treat. Of course, we start this project that time is not really trying to make vaccine, but we are more interested to build platform technology to identify potential so-called adjuvant. And of course, Sanofi is really leading in the field to making the vaccine against C. diff. They have antibody as well, whole toxoid in phase 3, and Novartis as well as others working on the C-terminal plus their own adjuvant, and some other people working on the synthetic peptide. And we are interested using lipoprotein. You remember talking about Dr. Ling already mentioned, using that as an adjuvant. And the focus is using the so-called receptor to block the binding of the toxin to the receptor, as well as another protein called toxin CDE. This is make a hole for the toxin to release. So if antibody can block here or blocking the toxin, then we can neutralize or protect people from the diarrhea. And we are very focused on the C-terminal, and there's a lot of repeat. So that's why we are interested in this, because they have a lot of repetitive sequence. And potentially will be a good adjuvant entity. So we clone the so-called C-terminus, and the receptor binding domain is 911 amino acid long. So we clone using a synthetic gene, and then clone into the vector, and then express them. Here, just show you the purify. Of course, there's some degraded material, and if you're using the specific antibody against the toxin, you can recognize the material we express. The first thing we check is this material have the biological activity or refold correctly? The one thing is the toxin can hemagglutinate rabbit cell. So we check, and indeed, they're able to hemagglutinate the rabbit cell. So we show they fold correctly. And then the other thing is binding. Maybe you cannot see well. Within 5 minutes, they will bind on the surface of the viral cell. And then 15 minutes, they will go in. And then 30 minutes, they will degrade it. So we sure we have the recombinant protein and express and refold correctly. The next thing is do immunogenicity, as shown in here, 30 microgram. This all without any adjuvant, just by recombinant protein. Okay, so highly immunogenic, and also show a balance of the so-called antibody response and also the subclass antibody response. The next thing is they can recognize the antitoxin or not, so we do the Western, and then they do recognize the toxin. The next thing is check the functional antibody and the the receptor binding domain. With enough dilution, they will block the so-called viral cell, because the toxin will kill the viral cell. If you have the antibody, they will block it. So here, just show you the titer. We make a couple of lot, and 3 microgram, 30 microgram have the neutralizing titer. And then the second one is better to have a 256 titer. The next question we'll say is, are they going to be able to protect the so-called the challenge in the mice? And in fact, you can see the 3 microgram around 20%, and then 30 microgram, the 90% in the lot 1, and then the 30 microgram, 100% protect the mice against the toxicity. The next thing we want to ask, because this 900 amino acids expressed is not good, because so we cut it into 3 pieces, and each one have 3 potential receptor binding domain. okay, because it is a repellent sequence, so-called carbohydrate binding domain, each one have 3 of them potential, and this whole receptor has around 7 potential receptor binding domain. The next string is cloned in the expansion system and then purified. Here to show you the purity of the material of the so-called F1, F2, F3 compared to the receptor binding domain. And then do the immunogenicity as well, do the neutralizing assay. We find the F3 is better than F1 and F2, even though they have some neutralizing activity, but definitely the F3 induce better immune response. As you can see, they're very high antibody response. This, again, no any adjuvant, just the recombinant protein. And then we test, because our goal is adjuvant activity, or the so-called, can they stimulate dendritic cell or activate dendritic cell. So just show here the so-called receptor binding domain in CD80 or F1, F2, F3. Essentially, all the so-called service markers, they can activate. And also, you will wonder, you did not using any adjuvant, they are so immunogenic. How about residual LPS in there? So we also put in the polymyxin B, and in fact, there's no difference between putting polymyxin B or just the recombinant protein. So all the immunostimulation or the immunogenicity is response by the recombinant protein. And also, it can show the so-called cytokine. And again, if you mix with the polymyxin B, there's no difference. So definitely, really clearly show all the subunit, including F2 or F1, they can induce a so-called activation of the dendritic cell. And the best part is the C-terminal part of the receptor. In fact, this is most of the other companies using it too as a antigen for vaccine development. So they can induce cytokine, and we want to know, can they activate other bystander type of the immune response? So we're using overwhelming, as shown in here. They are able to help the overwhelming enhance the immunogenicity. So here, just a summary, we have identified the so-called toxicity. It can be reduced by raising antibody against the receptor binding domain. And at 30 micrograms of the receptor binding domain, we can totally able to protect the mice from the toxin challenge. And also, they function as a toll-like receptor agonist. So in order to really protect as well as trying to reduce the dose, we combine so-called lipoprotein technology with our so-called receptor. As shown in here, this is a so-called without the lipidated, and then this is lipidated. Even the 3 microgram is higher than the 30 microgram. Definitely, there's a synergistic effect between the so-called lipidated with the receptor binding domain. The next thing is you see they can protect better compared to the receptor, just the receptor binding domain. The lipoprotein equal 30 microgram, 70% protected is non-lipidated, and then lipidated is 100% protected. The next thing we want to do is increasing the so-called challenge amount of the toxin and also reduce the dose to see we can repeat the experiment and also show the protection as well as the potency about the liporeceptor binding domain. As shown in here, 0.3 microgram, we have around 90% protection, and 3 microgram is fully protects 100% protection. So in this study also now we have receptor binding domain itself is adjuvant, but if you put lipidated part, they will have synergistic effect. We have the super so-called antigen for Clostridium difficile vaccine development. So I stop here for any question you have. Yes.
Does this RBG domain have any direct antimicrobial effects
No, they just bind to the cell. They don't.
Immunostimulation it provides, would it just give an adjuvant effect to protect against bacteria directly without an antigen challenge?
You mean they have antibiotics-like activity?
No, I'm just thinking that the adjuvant effect, can it be utilized as kind of an innate immune modulator against infection?
We have not challenged just by itself to see they will protect. But we are more interested to see the mechanism that time. And in fact, we did the mechanism of action is, in fact, they are toll-like receptor for light agonists. Because we can using the toll-like receptor for lockout mice as well MID88 lockout mice to show they are toll-like receptor for agonists.
How does your vaccine behave in a hamster model?
Okay, that's a very good question because if you're using the so-called using the bacteria in the spore and then challenge, not using the toxin to challenge, so-called the current lipoprotein A receptor binding domain is around 50% protection. But if you're using the toxin B receptor binding domain combined together, it's fully 100% protection at 3 microgram dose. You need the, similar to us, you need the toxin B receptor binding domain for full protection from the spore. Okay, but if you're using the challenge of the toxin, just the lipoprotein or the receptor will fully protect.
Okay, that's my second question. One last question.
Hi, great talk. Can you say something as to whether the subunit-based product allows you to cross-protect against other toxinotypes? So so how does the subunit work and the lipidated subunit work from the perspective of spread of the immune response against the receptor binding domain? Does it cross-protect against all toxinotypes?
You mean against the toxin B?
No.
They can induce the neutralizing titer, but around maybe 1/10 of the potency.
Okay, that's fine.